Okay, so the first question is asking about the chemoreception of smell and taste. So, basically, it's pretty simple. So, first, here we have, let me grab my, here we go. Here we have the nose, or this is the nose. And when we smell something, it goes in our nose, and about 95% of what we smell is trapped in like our nose hairs, because that's like we could smell like a virus or a bacteria or something. So it's trapped by our nose hairs, which we're super grateful for that. But the other 5% is trapped by the olfactory neurons, and these and then that is how we actually perceive smell. And so basically odor molecules get trapped in this part of our nose, and they get stuck on mucus, and then they get dissolved into these receptors. So like this is the nose, and then these are the like the mucus, and then here are the receptors, and here's the odor. So it gets dissolved into the receptors, and then it goes up to the brain where information gets processed. And so that is how we perceive smell. And taste. So here we have our tongue, and then here, these little things right here is the papillae, and these aren't taste buds. Taste buds are actually found within the walls of papillae. So this is the taste receptor. And each taste receptor, these aren't taste receptors. This is a taste bud. So yeah, people get confused saying these are taste buds, but this is a papillae. The taste bud is found within the walls of the papillae. And these little taste buds have about 100 to 150 taste receptors. And so the more breakdown of how a taste receptor looks is this part. So it has taste cells and microvilli, which is just like taste hair. This whole thing is the taste bud. And so, like this says, the tendons will interact with taste receptors through a number of different mechanisms to depolarize the cell. And so after the cells depolarize, it releases neurotransmitters that stimulate sensory neurons, and it goes up to our brains to process the information that we're tasting. And so we can taste 5 different things. First is salty, and salty is sodium and potassium is involved with us tasting salty, and then hydrogen or acids is how we taste sours, and then bitters is through calcium, and then sweets through carbs, and then umami is through glutamate. And so yeah, that is how we can taste. And yeah, so the other question, let me actually erase this. Okay, so how we hear. So the first thing is, yes, sound waves enter through our ear. I guess I can actually draw this. So here we have our ear, right? Or actually, let me draw it like this. And these are sound waves, and they go in through our outer ear, through the eardrum, and the eardrum is like about half the size of the size of a dime. And so once they reach the eardrum, so this eardrum, it starts to shake, or not shake, vibrate. And then because the eardrum vibrates, it makes 3 tiny bones vibrate, which is the malleus in this one, and then this one. I don't know how to pronounce them. And these things, they amplify the sound vibration. and then they send them to the coal. I don't know how to spell it. Pronounce it, but it's this thing. And this is how it looks. It kind of looks like a snout, and it's filled with liquid. And once sound is entered, it literally makes the liquid like ripple, kind of like throwing a pedal into a river. And yes. So the stereocilia sits on top of the hair cells, and they're grouped together as hair bundles. And so this is kind of how it looks. And that makes okay. Yeah, we have these hair bundles, right? And when the sound waves cause the fluid to ripple, it also moves the hair bundles. And so that movement causes electrical signals. And as the hair bundles are moved, ions right here are rushed into the hair, causing the release of chemicals. So ions move into the hairs, down the hair cells, and release chemicals at the bottom of the cells, which are neurotransmitters. So yeah, ions first go through the hair bundles, down the hair cell, and then release chemicals, which are neurotransmitters, and then they connect to the auditory nerve. And then they create electrical signal, which is then sent to the brain. Okay, yeah, so there's different types of hair cells that respond to different frequencies. And so these are just a few of them. And then and then the last step is that the auditory nerve carries the electrical signal to the brain, and then the brain interprets the message as sounds, and that is why we recognize and understand what's going on around us. And so yeah, and then so focusing light in the eye. I put ear, but it's meant to say eye. Okay, so yeah, the cornea and lens, that is just how light enters the cell. So light enters the cell through the cornea, which does the focusing process. Light then passes through the aqueous humor, reaching the lens. And so the ciliary body. So this is kind of how it looks. So we have this thing, and then we have these things right here, and that represents these things. And so I can actually draw this. I'm going to draw it with the thing. So this is the lens, and then this thing right here is the ciliary body, and then these little things is the suspensory ligament. So the ciliary body is the ring of muscle that surrounds the lens. So as I drew it here. And then these things are the suspensory ligaments that just connect to the ciliary body to the lens, and then all this together either makes this bigger or smaller. And so, yeah. And then we have photoreceptors. So I guess we can draw that. I'm just going to erase this. Okay. Let's make a thing here. So we have photoreceptors, and this represents that. So first we have rods and canes. So the rods allow us to see light. Cones allow us to see colors. And so the rods are super sensitive to light and are and then the cones are less sensitive to light. So the conversion of light to electrical energy is just when light hits the photoreceptors, it causes a chemical change in the pigment within the rods and cones that changes and activates a cascade of molecule events, and then leading to hyperpolarization of a photoreceptor cell. And then neurotransmitters are released, which causes a membrane potential, and then an action potential is caused. So first we have the photoreceptors, then we have the neurotransmitters, and then we have an action potential. Oh no, we have a membrane. Then we have an action potential. And then we have the on and off surround. Okay, so when this is yellow, let's say, when this is yellow, that is called on center. When this is, let's say, gray, it's considered an off center. When this is gray, that's considered an off surround. When this is clear, it's considered an on surround. I'm just gonna erase all of this. Okay. So they are activated when light hits the center. And then off surrounds is these cells are inhibited when light hits the center of their receptive field and activated when light hits the surround. And so yeah. So basically the summary is the cornea and lens focus light into the retina, and photoreceptors, rods and cones in the retina convert light into electrical signals through phototransduction, which are then transmitted to the brain. Centers around perspective fields. This organization helps detect, construct the edges, enhancing visual perception. So this allows us to see all of this allows us to see shades. And so yeah. And that's all.